Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. We're thrilled by the overwhelming support for our episodes featuring top-notch experts delving into various biological realms. Thanks to the feedback from higher biology students, we're launching shorter, punchy revision episodes. Get ready for quick summaries of key areas to turbocharge your exam prep. These short episodes can support your exam preparation and are not intended to replace other important aspects of your revision. In this short revision episode, we're jumping into the fascinating world of gene expression, a fundamental process in understanding how living organisms function. Genes are sequences of DNA which carry the coded instructions to make protein. Only some of the genes present in a cell can be expressed after the cells become specialised. Gene expression involves transcribing DNA into RNA, followed by translating the sequence of bases in the RNA into amino acids, which are then used to make protein. Transcription and translation involves three types of RNA: messenger RNA, mRNA, transfer RNA or tRNA, and ribosomal RNA, rRNA. RNA, which stands for ribose nucleic acid, is a single-stranded molecule consisting of RNA nucleotides. There are 4 different RNA nucleotides based on the 4 different bases: adenine, uracil, which replaces thymine, cytosine, and guanine. Messenger RNA carries a copy of the sequence on DNA from the nucleus to a ribosome. Each triplet of bases on an mRNA molecule is known as a codon. Many candidates find describing the structure of tRNA tricky, so remember to include that a tRNA molecule is folded due to complementary base pairing. At one end of the molecule, there is an amino acid attachment site carrying a specific amino acid, and at the opposite end, there is a triplet of bases known as the anticodon. Ribosomal RNA and protein form a ribosome. Expression of genes leads to the synthesis of proteins using the sequence of bases on the DNA. And the first stage of that is transcription. Transcription occurs in the nucleus of eukaryotes. It involves the enzyme RNA polymerase. RNA polymerase moves along the DNA, unwinding the double helix and breaking the hydrogen bonds between the bases. RNA polymerase brings RNA nucleotides to line up opposite complementary bases on the DNA. A pairs with U and C pairs with G. RNA polymerase synthesizes a primary transcript of mRNA from RNA nucleotides by complementary base pairing. Now, many candidates fail to fully describe the function of RNA polymerase, so remember, it unwinds the double helix and breaks the hydrogen bonds between the bases, as well as adding complementary nucleotides to the primary transcript. During transcription, DNA sequences which do not carry the code to make protein or are non-coding are transcribed along with coding sequences. The primary mRNA transcript therefore contains coding and non-coding sequences. Those non-coding sequences are called introns and the coding sequences are called exons. RNA splicing involves cutting out and removing introns from the primary mRNA transcript and splicing together the exons. During RNA splicing, introns are removed and exons are spliced together to form a mature mRNA transcript. The order of the exons remains the same. Alternative RNA splicing allows several proteins to be produced from a single gene. During alternative RNA splicing, different combinations of exons are retained and spliced together to form different proteins. After production of the mature mRNA molecule, the mRNA leaves the nucleus through a nuclear pore and attaches to a ribosome, where translation occurs. Different mRNA transcripts can be produced from a primary transcript, depending on which exons are retained. The next stage of gene expression is translation. Translation occurs at the ribosome, and it begins at a start codon and ends at a stop codon. tRNA is involved in translating mRNA into a polypeptide at the ribosome. As the ribosome moves along the mRNA, anticodons on the tRNA molecule line up opposite complementary codons on the mRNA and bring a specific amino acid into the sequence. The tRNA molecule leaves behind their specific amino acids, which are linked together by peptide bonds to form a polypeptide. The sequence of bases on the DNA determines the order of bases on the mRNA, and this determines the sequence of amino acids in the polypeptide. Gene expression is like the conductor, orchestrating the symphony of life. Understanding this process not only unlocks the secrets of how cells function, but also holds the key to unravelling the complexities of genetic disorders and diseases. So hit subscribe on whatever platform you get your podcasts, so you don't miss the rest of our short summary episodes, nor our regular episodes with leading experts. And remember, this quick summary is intended as just one element of your broader revision plan. There are lots of resources available via Glow, such as Scholar or WestOS, as well as BBC Bitesize. Or why not check out one of Lecky's great revision books and use the code LeckyPodcast40 to get 40% off on their website, www.leckyscotland.co.uk.